So this work is going to, it's going to ask a question. Now to ask the question, we need to set the stage for the method. So I'll first talk about the method that we developed. And then after that, I will talk about, does that method do what we want it to do? And why an in silico imaging trial for that purpose? So these are exciting times, and there's a lot of excitement in this room as well as we develop new imaging methods for radiopharmaceutical therapies. There are new strategies being developed, new systems being developed for optimization for, new systems being developed, work by Robert Mioka, some work in at WashU by Linksci, Dr. Yuan Chantai, with whom we collaborate. New image analysis methods being developed, new quantification methods being developed. So there are all of these wonderful methods that we are developing, all of these new imaging technologies that we are developing for improving quantification, improving visualization of radiopharmaceutical therapies as they get inside the body, as they emit the gamma ray photons. And so with all of that, and I understand, I believe all of you know the imaging context here, right? With radiopharmaceutical therapies, you have some alpha beta emission, and then that actually also emits gamma photons. And these gamma photons can be imaged, so SPECT provides a mechanism for being able to perform the imaging. So I'm assuming that background, but again, so the key question then becomes, how do we optimize and validate these methods in ways that are clinically relevant, right? So of course we can do, physical phantom studies, we can do simulation studies, but how do we know that we have obtained evidence that is clinically relevant, such that we can then sort of get the early stage data that motivates us to do an actual clinical trial for these therapies, and that provides potentially data that can help us guide the clinical trial that we want to do as well. And in some cases allows us to do things which a clinical trial would not be able to do, which is what I'll talk about today in the in silico imaging trial that we had performed. So, but like I mentioned, this requires me to provide some background on the method itself. So I'll first go through the method. So again, I think all of you are familiar with this. I will just go through this slide only for context here. We have some activity uptake, we have a SPECT system that gets some projection. And the idea is that we use this projection to quantify isotope distribution, right? Now, there are several challenges, particularly with alpha particle radiopharmaceutical therapies, that make this quantification task very difficult. As you can see, with your conventional SPECT images, the images are like still noisy and low resolution, but with alpha particle SPECT, it becomes even more noisier, much more noisier, in fact. So even compared to diagnostic count levels, alpha particle RPTs can have 20 times lower count levels. So the images are highly noisy. There's also this issue of stray radiation related noise. So when you have such low count levels, even things like, count from cosmic radiation, patients sitting in another room, all of this starts also having an impact. And this we learned the hard way from actually doing a physical phantom study, and like, what are these counts? We don't see them. Where are these coming from? And turns out because the number of counts are so low, we just cannot ignore these counts anymore. Then, of course, there is complicated isotope physics. I don't need to describe to you any of that to you all. And then, in addition to that, there is complicated SPECT physics. There is attenuation, there is scatter, there is limited system resolution due to the collimator, to the detector, and so on. So as a result of that challenge, it becomes very challenging to perform quantification. Now, the typical strategies for quantification revolve around, let's take the projection, do a reconstruction, and define some kind of masks on the reconstructed images to quantify activity uptake within some volumes of interest. I'll refer to that as VOIs. Now, the situation here is the following. We have projection data where the number of counts are extremely low. Meanwhile, the reconstructed images, if we are trying to do reconstructions, we are typically having 128 or 128 pixels in a slice, and maybe like 128 slices, sometimes 64 slices. That's a pretty large number, like very large number, right? So in terms of mathematics, the way I would frame it is that you don't have enough data to be able to estimate activity uptake values in all of those voxelized bins with very high precision. And as a result of that, and this is again with conventional analytical based reconstruction methods, what we see is that even fine-tuned analytical reconstruction methods yield high values of bias and standard deviation in estimated activity uptake. And therefore, there has been a lot of research in this area on how to improve this, including work by Luke and your group as well in looking at Monte Carlo based reconstruction methods and the promise that they are showing. Our approach was, what's the task here? So if the task is not necessarily looking at voxel based dosimetry, but instead mean activity quantification, then the reconstruction step is sort of an intermediate step towards the goal. So we asked the question, can we skip reconstruction? And the challenge is that we are trying to estimate all of these voxel values, and all of these voxel values are eventually helping us get something. Why not just skip that intermediate step? Can we go directly from the projections to the mean activity uptake in the VOIs? So that was the key idea. This goes back to some early work in the 1980s by Rich Carson on an ML ROI based approach, and we sort of expanded that approach, advanced it in the context of radiopharmaceutical alpha particle RPTs. The idea was that from when we do these scans, we also acquire a CT scan in conjunction with the SPECT scan. And with the CT scan, we can get VOIs, volumes of interest for the organs, and some cases for the lesions. And with those organs of interest, we can then define sort of VOIs. And what we can do is, instead of reconstructing activity at a voxel level, we can reconstruct activity at a volume of interest level. So let me just go through that math a little bit. This approach led to a low count quantitative SPECT method, which we call LCQ SPECT method for alpha RPTs.
If you want to do tumors that you cannot see on CT, can you do SPECT first?
Yes.
And then
Yeah.
put it and then go back and then.
Yes. So
you do recon once, you define the ROI, but then you go back and
go back. Yes. So I like to think of this approach as sort of complementary to reconstruction-based approaches, because there are lesions that we are not able to see on the CT scans. So what you can do is do a SPECT reconstruction, use that to define the VOI, and then use this approach. And from the data that I will show to you, you will see that this approach does lead to very reliable activity of estimate. So if the goal is quantification, if you can define the VOIs, you can do a very good job at activity quantification with this approach.
So you should do recon for visual, but then for quantification, you skip.
For visual and for delineation if the lesions are not visible. Yeah. Or more specifically VOI definition for those lesions which we cannot see. So to just to provide some mathematical basis for this, here is your object F, the radio tracer distribution, your imaging system H. This psi denotes the radiation related noise, or the mean of the radiation related noise, and this N denotes the noise itself. And so this H, as I like to call it, maps from our continuous patient to a discrete detector. And to specify this in the context of image reconstruction, what we do is take that continuous patient and voxelize that patient, right? And so this becomes a matrix that relates a voxelized patient to the discrete detector. Now, you can take voxels, but what you could instead do is define VOIs, such as lesions, such as gut, such as bone, and call each of them as having its own mean activity uptake defined by lambda, right? And so you can have a system matrix, and this system matrix now relates not a voxel to a detector pixel, but a VOI to a detector pixel. And so that's the underlying, overall math behind this. What details I'll refer to your paper, where we talk about more about how we derive this particular system matrix. It's still a matrix, but it's a much more low dimensional matrix, because when we think about reconstructions, we are doing maybe 128 cube voxels. When we are thinking about doing this step, we have maybe like 20 regions to estimate, maybe 30 regions to estimate. Sometimes patients may have a lot of systemic disease. So in that case, you may have many, many lesions, but still the number will likely not go to 128 cube. And so we have this imaging equation. We know that this noise is Poisson distributed. We are able to also model this psi as radiation noise also having Poisson distribution. We are able to measure it using blank scans. And then what we do, we maximize the likelihood of lambda given this acquired data, and that gives us an iterative equation. Using the iterative equation, we are able to estimate the activity values in different regions of interest directly from the projection data. And so I'm going through this a little bit quickly because I know all of you are excited to see what the VIT and I'm excited to present it too. But if you have any questions, feel free to stop me, and I would be happy to answer. So what we did was that we did some simulation studies with these patients with XCAT. We took a patient, we had some lesions inserted. We then allowed this image this patient virtually on a SPECT system. This was a G640 system. We reconstructed that using an OACM based approach. Based approach with partial volume effect compensation and our low count quantitative SPECT approach. And then we quantify performance on accuracy, on precision, as well as overall error. Remember, this is just for a single patient that we're talking about here. And so what do these results look like? So what we are seeing is across different sizes of the lesion, you can see the lesion diameter here. Here's the normalized bias, and here's the normalized standard deviation. Our method shown here in green gives close to zero bias in estimated activity uptake and gives variance that approaches what is called as the Kramer-Rao bound, which is the theoretically lowest value of variance for any unbiased estimator. So if you don't know what Kramer-Rao bound, it is, think of it as, the absolute best that you can do, because you have noisy data, you cannot get zero precision, right? Zero variance, you'll have some variance. The Kramer-Rao bound tells you that if I have an unbiased estimator, what's the best variance that I can get? And that's what we see is that with our method, we get variance that really approaches the Kramer-Rao bound. Now, the same is observed across contrasts too. Again, we see that we have different values of lesion to bone uptake ratio. Our methodology yields close to zero bias and outperforms the reconstruction-based methods. Specifically, when the sizes are low or that the lesion to bone uptake ratio, the contrast is low, our methodology does very well compared to the reconstruction-based approaches. And also in terms of normalized standard deviation, it does very well.
Shouldn't you be gaining some bias because you're assuming organs are uniform?
Yeah, so these studies have been done under the assumption that, under the imaging system model that, not the imaging system, the object model that the activity is constant. So we have studied performance when you have heterogeneity in the activity uptake. And what we find is that up to a certain level of heterogeneity, this method continues to outperform reconstruction. But after a certain level of heterogeneity, the performance deteriorates.
Can't you assume it's uniform only for that region of interest, but then for everything else is voxelized?
Yeah, so there are adaptive approaches that could be taken. And I think that's another exciting area is, if you can, again, do an initial reconstruction, and you could say, oh, there is this area which seems almost close to constant uptake, but there are other regions where the activity uptake is more heterogeneous. If I could define VOIs that approximately look homogeneous, then through this methodology, we will be able to estimate activity uptakes in this VOI. So for example, let's say the liver, I'm trying to estimate activity in the liver. Liver may not have constant uptake. There will be regions which have high and lower uptake depending on the lobes that you are in. And so with this methodology, if you are able to first do a reconstruction and look at that heterogeneity, then you can define VOIs. And with those VOIs, you may be able to, if those VOIs within that liver are homogeneous, you will get accurate activity uptakes within those regions. Whereas, yeah, so I think of this as really like a complement to something that complements reconstruction-based methods, where this is set up for the task of quantification, provided you can satisfy the model assumption of VOI uptake almost being constant.
Specific to the system.
The Kramer bound is specific to the system and to the thing that you're trying to estimate. So in this case, what I'm trying to estimate is the mean activity uptake. So I'm asking the question, how well can I estimate the mean activity uptake given a log likelihood model that is based on the Poisson noise?
Right? I guess just with the small diameter lesions,
yeah,
the Kramer bound is higher than variance you see in the OCM. But I would expect that way. Yeah.
So if you look at the smaller lesions, right, what happens is that the activity uptake also is lower in those lesions just by virtue of the fact that they are small, they don't have as much activity. So again, it's Poisson noise, the noise scales as the variance scales as the mean. And so that's an intuitive way to think about why the variance would be higher. Another easy way to think about this is take your iPhone camera, take it out in the night and take a photo and you will see graininess, right? Kramer bound would be higher there as well.
But OCM is not strained by Kramer in this case.
So because it's a bias estimator and it's
so
not unbiased?
So you will note that at the lower lesion diameters, right, the OACM actually gives better normalized standards. This was going back to the question that I asked you. Yeah, that I asked of you, that there is a bias variance trade-off, right? So I would say that the Kramer-Rao bound for reconstruction-based methods would be different from the Kramer-Rao bound for the our method.
So that would be higher because you're estimating less, like you were saying,
yeah,
compared to lower because you're estimating less amounts. Like you're up with small amount.
Sure. So what I want to clarify here is this is the Kramer-Rao bound when you are estimating mean activity uptake, right? So the model is set up for that. Now, if you think about reconstruction has bias. So Kramer-Rao bound is not even the right metric to look at. There is work done in the area of this biased Kramer-Rao bound, which accounts for the bias and then derives the Kramer-Rao bond for that. So I therefore do not want to contrast Kramer-Rao bond that we have derived with the Kramer-Rao bond of the OSCM-based reconstruction.
So the Kramer-Rao bound is the lowest variance, theoretical variance for an unbiased estimate.
That is correct, yeah.
But these techniques are actually biased.
The
Therefore,
reconstruction.
They could have
Yeah.
lower.
Yes, yes.
How many cases do you have?
So this one was, we are doing Kramer-Rao bound, so this was multiple noise realizations, and this was, I think it was 50 noise realizations that we had considered to derive the empirical estimate of the variance, and the Kramer-Rao bound is not, it's theoretical, so you don't need to do any kind of.
It's for single case, right? Not multiple cases, average of multiple cases.
So this, like I mentioned, is a single patient, but you have doing multiple noise realizations. So you're doing multiple, think of this as taking the same patient and scanning them 50 different times.
And did you compare it with other, for example, patients?
That's why the in silico imaging trial.
So you're going to show us.
I'm going to show you, yeah.
So how to model the scatter estimating the ocean? You are outlier.
Yeah, so I'm glad to see all of these questions. So the way we do it is, it's a system matrix is obtained through cement. So this H matrix, this H matrix that you're seeing here, the way we obtain this is H matrix is that we have a VOI. You define the VOI from the CT scan, and then you run a cement simulation, and that tells you the system response for a certain VOI. And you do the cement simulation for a very large number of counts, so you get approximately noiseless projections, and then divide system matrix.
So you store this.
And then you store the system. So this is pre-compute and store.
So for that figure that for the last page you showed,
Yeah.
What's the iteration number you compare for? It should be the fixed iteration number compared to.
So we did convergence studies actually, and we identified that, and so these results that I'm showing you are obtained after the methodology actually converged to a certain value. So we had to do several thousand iterations for that purpose. Because what we wanted to ensure was that the convergence was happening across all the volumes, all the sizes, and so on. So we, again, in the paper, we have the data that shows how we had to convert, how we did the analysis to identify the number of iterations. And from what I recall, it was a very large number of iterations.
Because I find, especially for the small numbers, even if you're using contour for iteration, it's still not convergent.
Yeah, so we keep pushing. We do more iterations and eventually we find convergence. And I think it's a very important thing that we ensure that there is convergence before we actually make some. Now, I cannot comment on the. So we try to be as rigorous in our experiments as possible. So for our reconstruction-based methods as well, the results you're seeing were optimized because we wanted to ensure that our reconstruction-based methodology is also the best of what it can be. And of course, our methodology is the best of what it can be. So we are comparing apples to apples here. Yeah. So this is good. And so, of course, it's simulation. So we did some physical phantom studies too with some anthropomorphic vertebrae kind of phantoms and NEMA phantoms. And again, we observed that our methodology shown here in green continues to outperform the OSCM and GTM based methods. Now here you will see not close to zero bias. This is what I'm showing you here is not the bias, it's the error, which is a combination of bias and the variance. But these numbers are on the higher end, which shows that when you're going to apply this technique in practice, then you are going to have to deal with issues like perfect UI alignment and so on and so forth. So all of this is good. It gave a lot of promise. Seems like it's outperforming. Seems like it's giving very good accuracy. Seems good, very good precision. All of this is exciting. But then we had the question, how about its performance across multiple scanners? Because this was just a single scanner. No, but why should I care about performance across multiple scanners? Now, if Carlos would be here, he would tell you the answer. But since he's not, I'm going to try to do that. Or Arman can help answer. So different centers, and you have a GMI, we have a G640, we also have a 670, and now we're getting a star guide, and you are getting a star guide, and there are those which have the Siemens Symbia, right? So different scanners have different centers, have different spec systems. This leads to variability. If you have variability in the activity uptake that is measured, so given a certain activity uptake, you have variability in your own estimates of the activity uptake, this would lead to variability in dose quantification. This can then hinder robust dose-response relationship prediction. And then this can complicate combining and comparing data across different centers. So therefore, there is an important need to assess reproducibility of our low count corner dystrophy method across multiple scanner configurations. Right? So we want to be able to assess how well our method works, not just for one scanner, but across a range of scanners. Now, one way to do this would be to do a real imaging trial. Take a patient and tell them, go to Vancouver, and a lovely place, you'll enjoy here, and then come back to maybe St. Louis, and then come to Johns Hopkins and go to USCF and so on, and then get all your scans done, and we'll assess reproducibility. So of course, it's very expensive. It's also very time consuming for the patient as well. The patient gets much more dose, so there's a safety issue too. There's all kinds of ethical questions that arise due to that. There's a lot of logistical questions that arise due to that. And there's a bigger issue. There's no ground truth. So how do you know how accurately you're doing things? Why did we do simulation and physical fandom studies? Because we know the ground truth there. So there is an issue of lack of ground truth as well. So that brings us to being able to do this in a virtual setting. So where you have a virtual population, you take sort of a set of patients who are clinically representative and represent the kind of population that would be imaged with these therapies. You do virtual acquisition, so everything happens in the lab. You don't have to take the patient, this virtual patient, to any scanner. You want to do this very accurately. And then you do virtual interpretation. And there's been a lot of work in virtual interpretation using things like model observers, which I also presented yesterday about, and their role in being able to quantify detection performance. But here we try to push that towards quantification. How well can we have a virtual interpretation that is quantification based? So virtual imaging trials, as you can see here, they are not as expensive. Basically, you're using GPU power or CPU power. They are certainly safer for the patient because there's no patient getting any radiation dose. It's logistically less challenging. You have to just be in your lab, don't have to go anywhere to do these studies. It's safe. And very importantly, there is ground truth that is available. So you can know how well your method has performed because you know what the truth is. Now, all of that is good. But at the end of the day, I like to see them as something that provides motivation and for an eventual clinical trial as needed, if it can be done. So I like to think of these as early stage evaluation studies. Once we do these early stage evaluations, if the results show promise, then this motivates doing evaluation of patient data, maybe in prospective settings as needed. So for our setting, conducting the evaluation that I talked with you about, where we are trying to assess performance of the methodology across multiple scanners, this would be very challenging, costly, expensive, time-consuming, logistically challenging, and so on. So we said that can we do this in an in silico setting? And that led us to ISIT QA. So when you design a trial, what do you do as the first thing? You give it a name, right? So we gave it a name, ISIT QA. That's actually not the first thing, but
so it's very important, by the way, like the way you also title your papers and you sort of talk about these clinical trials, and I think these are extremely important.
Yeah, I've noted that when I've heard this actually. And one of the things that we're trying to do here was really try to emulate a real clinical trial. So when you go to like an ECOG-ACRIN meeting or any kind of ASCO and so on, right? You see that the trial has a name, right? And that name actually, I've heard this, that a trial with a good name actually can get more recruitment. So we don't have to worry about recruitment here because we recruit more. But it gives, and I like this ISIT name, and I should also acknowledge Dr. Dan Thorix, who is a collaborator, for initially suggesting this. But I kind of really like this whole ISIT thing, which we have then gone on to a metaverse where we have multiple ISIT trials. So what do you do? What's the next thing you do when you have a trial? You have a primary objective. So you define a primary objective. Right? So evaluate reproducibility of this methodology, LC-Q-SPECT, across SPECT scanners and collimator configurations. So when I discussed with Arman about this, the sense I got, Arman, from you was that you kind of wanted to hear our experience and how did we go about thinking about designing this trial, right? What was our philosophy? What was our study design? And how did we think about study design? So that's kind of what I will try to do in today's talk is, trying to tell you what were some constraints that I had, what were some challenges that we faced, and it's more of a story that I will try to share here. So we first define a primary objective. You evaluate reproducibility of LCQ SPECT across SPECT scans and colorimeter configurations, right? So, and I'll go into details on this. So let me show you the overview of our clinical trial design. Our clinical trial design, as I showed earlier, in terms of the broad schematic, consists of three broad things, which is generating the virtual patient, so your virtual patient generation, doing the SPECT imaging of these patients, then performing data processing and evaluation, which I will call them as, in a single step, as sort of doing the clinical task. The trial has a primary objective. In this case, the primary objective is to evaluate reproducibility of the method across different scanner colorimeter configurations. But trial also has secondary objectives, and our secondary objectives included evaluating the accuracy of the method. So reproducibility is good, but we also want to evaluate how accurate our method was. Evaluate test, retest repeatability. Now, for those of you who may have done these test, retest studies, right, coffee break studies, as we also call them. So we also try to do like a test retest, where you take a patient, you scan them, and after that you tell them, stop for a certain amount of time. Our patients don't have coffee, so we don't have to spend on coffee too. And then you again retest them, and then you see how the method works out in that situation.
What do you say, test retest?
Oh, it's a secondary object. It's a secondary object. Yeah. And again, I'd like to acknowledge Dr. Dan Thorek here for some of these creative ways of thinking about this.
That's exactly the kind of thing that needs to be done. It's very nice. Yeah, just like in any clinical trial, you have primary objective.
Yeah.
Even when you write ethics, I guess you call them RABs here, and you call them IRBs in the US.
Yes.
It's like you think about primary and secondary aims, that stuff.
Yeah.
So
we got an IRB for this as well.
So tell us
It
about
was an
that.
IRB.
This was the first time I found out. I was like, what on earth? So we call these research ethics board meetings,
Yeah.
REB. I guess in the US it's Institutional Research Board.
Yeah.
IRB.
Yeah.
Basically, when we conduct, let's see who knows about REB. Nargis, you know. So what's your interpretation of what an REB is? Tell us. What's an REB?
One.
It's Research Ethics Board.
Yeah, and I wrote some
Mhm.
It was by project,
Mhm.
and then
Yes. And the IRB, in the US, we call it the IRB, Institutional Review Board, and they want to ensure that your trial is justified, it's safe for the patients, because there's a risk-benefit analysis, and then they do the risk-benefit analysis and see whether it can actually provide endpoints that justify the risks that the patients have to take.
Yeah, because there could be a leakage of private information, all that stuff. So there's a risk whenever you access patient data, you have to justify to the ethics board that that risk is justified. So, but what does it even mean to do a ethics research?
Trying to emulate a clinical trial.
But what does it mean?
So we approached the IRB and we said, we are doing the study, and they were like, yeah, that's fine, do it.
No. What are you asking permission for?
They gave us an ID actually. We are not exactly asking permission. We are just trying to follow the same process as we would follow for a clinical.
Because I noticed that
So it's probably a bit of a stretch.
I know, but I like that stretch. So you're really putting yourself in that mindset.
Yes,
Going
exactly.
through that whole procedure.
Yeah.
You sought permission basically to
Yeah.
run this and to do evaluation.
Yeah, we got very excited about this. So I actually did a form, and I called up the IRB office, and I'm like, guys, we are doing this. We need to do this like we are doing a clinical trial. Our plan is to actually do a clinical trial also later on. So I reached out to them and I said, you have done this. And they're like, yeah, that's okay.
It's funny because you run away. I like to run away from all these RMEs. You run towards them.
I don't know if I like to run towards them, but I thought that this would be an experience in learning how to run towards them. So yeah, so
you did this number of patients. Because
excellent question, right? So as we conduct this in silico imaging trials, one key thing I would say is to ask, what is your primary objective? Any clinical trial, the primary objective guides the sample size, because you do a power analysis based upon your primary objective, right? And so you want to be very careful in how you are defining the primary objectives such that you have adequate number of samples to be able to answer the clinical question that you are trying to answer. So what is the most important question here that we are trying to answer? Is interscanner colorimetric configuration reproducibility, right? So we are trying to answer the question, how reproducible is our method? So we come up with a figure of merit that defines this reproducibility, and we came up with this intra-class correlation coefficient. And we asked the question, and this was actually a pretty long experience, right? You come up with ICC as a metric, intra-class correlation coefficient, and you are like trying to assess what are the ICC values which would be considered good enough so that the methodology is reproducible. And then you do pilot studies to be able to answer the question that, what is the kind of samples that I need so that I am able to, if the method is reproducible such that it has an ICC greater than 0.7, you would be able to see it. So you are actually trying to see if the statistical effect is possible or not, if there was one.
So to arrive at this number 280?
Yes.
Yeah, it does say based on power analysis.
Based on a power
I
analysis.
just noticed that.
Yeah.
So for other people, like when we write grants, usually we're saying, okay, give us money so we study like 100 patients. They're like, why 100? Why should I pay you for 100 patients? So we do power analysis based on very preliminary data.
Yeah.
We do some statistical calculation saying, if I want to show something with this much certainty, that's what power analysis is. There's alpha and beta and all those things.
Yeah.
So you did that,
You did that
again,
yeah.
using preliminary simulations
yes.
to do your comprehensive.
Yeah. And so based on that, we came up with this number of n equal to 280.
Really emulating.
Yeah, so really trying to emulate the clinical trial here, like, okay, 280 is the number. Now, in a clinical trial, and I think I'll maybe talk about this. So let's see if I can, yeah.
Because you try to do something virtual.
Yeah.
But you have exclusion criteria.
Yes, absolutely. Yeah, you have to have exclusion criteria, right? In a clinical trial, you exclude people based upon certain.
Do something that you exclude the data. So.
Yes, you have to be very truthful to this trial. This is a prospective study. You just start with what you do, and then you let the science take over, right? And then you don't go back, right? So, and so then, the data is what it tells you, and then you sort of eagerly wait for the data, biting your nails and seeing whether it hopefully everything works out. And thankfully, in this case, it did, right? So
you predefine your exclusion criteria, which would be like, do you recall on this one what it was?
Yeah. So of course, so let me go through the virtual patient generation, right? So what we did is we simulated patients with bone metastatic castrate-resistant prostate cancer. These were virtual patients who were treated virtually with radium-223 based alpha RPTs. So now here is the digital map of the virtual patients. These were generated using the XCAT Phantom. And here I'd like to point out, like, it's a virtual setting, right? So there are things that you can model and there are things that you cannot model. Now, in our study, we were able to model things such as height and weight of the patients. So we were able to obtain this from CDC data. Center for Disease Control in the US publishes demographic data for the height and weight of patients within a certain range. We were able to obtain a distribution of lesion diameters. These were radium-223 based studies, and there were clinical data that were available out there which told us what is the size of the lesions that are there. And so we had a distribution, and so we were able to find out what are the, we were able to get a stochastic variation in the sizes of the lesions. We were able to also find out data which told us that where in the body are these lesions present, again in a stochastic way. So we were held by these references which provide us distributions of probabilities of which part of the thoracic region or which part of the body actually contains these lesions. Details of all of this are actually there in our JNM paper that got published. Now, but there are things we cannot model. Like, we did not know what the PBPK parameters of the lesions were. So we kind of said, well, we don't know that. So what we can do two things. One is that, let us try to simulate some PBPK parameters. But we thought, well, our question is really activity uptake quantification. So can we just simulate or emulate lesions where we know the activity uptake, not worrying too much about the exact PBPK values? Again, this is in the context of alphas. So with alphas, even single time point dosimetry may be enough in some locations, in some cases.
So our job would be a bit tougher simply because there's probably more literature out there for how much counts there are in tumors versus what is the pharmacokinetic parameters.
Yes.
We're trying to look for these numbers.
And then we landed into an obstacle. We wanted to simulate heterogeneity in the tumors, right? And again, there's no data that tells us what is heterogeneity because alpha, and if you see some of these images, you will see they're just blobs, right? So there's no way for us to tell what is heterogeneity. So again, we had a design choice to make. We could arbitrarily introduce heterogeneity and say, okay, that is a heterogeneity in the patient. But then we can go all the way from being fully homogeneous to being fully heterogeneous, right? Or we could just say, let us specify a value of heterogeneity, specify something and work with that and realize that that is a limitation of the study, and we state that as a limitation of the study. So in our case, we assumed that there was no heterogeneity. All the VOIs had homogeneous uptakes. So again, these are kind of the choices that we have to make because we know things and we don't know things. And this is going to happen really in any virtual imaging trial because you don't have the actual clinical trial. So you would have to make some assumptions. But the good thing is, when you make those assumptions, you're able to specify those assumptions and say, within the limitations of the study, these are the results that we have gotten. And so that kind of helps you draw a claim, which is again grounded in rigorous science and grounded in the limitations that are provided. So in fact, I like to think that with RELAINCE, the claim idea we proposed actually is a very general idea, and it should really, it really holds across a lot of scientific literature too when we are evaluating different imaging methods. So again, sharing with you some experiences on how we went about doing these things. Then of course, there was also the question of, how much activity uptake should be in different regions. And again, we had some literature, some data that helped us get the mean and the variance in these activity uptakes, the different VOIs. And so here is a representative virtual patient. You can see the activity map on the top and the attenuation map at the bottom. So we gave it a number, patient number 78. We didn't give it a name because of PHI considerations, of course.
Well, if we gave it a name and then you anonymize
it. So now comes the second part, like, doing.
So the background, everything is constant, but the tumors you're varying.
Yeah, the tumors I'm varying. We have different.
The anatomy is varying.
Anatomy is varying.
The uptake of the anatomy is not varying.
No, the uptake, as in the uptake in the different physiological regions.
Standard deviation.
Yeah, we have different standard.
This is like you're assuming Gaussian distribution.
Yes,
and you randomly sample.
Yes, we randomly sample, right? So we have a large number of patients. For every patient, we assign a certain number of tumors based upon this probability distribution. We assign the location of the tumors based on this probability distribution. And then we assign activity uptake to tumors and to the different regions such as background bone, small and large intestine based on this distribution.
So James, yesterday Abhinav was asking me, when we do sampling, is it a constant activity or is it like a Gaussian distribution we sample from for the K values or pharmacokinetic?
We use log norm distributions for a lot of them, but Gaussian was something like for height and weight, for example. But something we're working on right now, which we're trying to figure out the best way to do is what if parameters are correlated. So are these all like marginal distributions?
Oh, that's a great question, actually. So in this case, we have these distributions are not, we did not have the data to be able to quantify the covariance.
And I think probably for these values anyways, the correlation would have less effect, but for our pharmacodynamic parameters, I think it's
it's like
that's one thing we're trying to get limited data, but how to deal with correlation and stuff like that.
And this is going to always be a challenge with VITs because if you knew everything, right, why would you do a VIT? Right?
Yeah.
In the sense that
you haven't paralyzed yourself by saying, oh, I don't know this, I don't know that, I don't know this, therefore I'm not going to do it. Like you make your assumptions, you make a conscious decision and you move forward. You move forward with that. Because in some sense, there's an early stage evaluation study and you get the data and then that data supports the actual clinical trial. Now, when the clinical trial happens, then you get all the data that you can get with that sample size that you have. But if you have to wait for the actual clinical trial, then then that's too long and it's too expensive. And with that, we are, in some sense, I like to think that we are subjecting patients to things that we don't know whether they will hold or not. But with virtual imaging trial, you get that confidence that yes, this thing should work. Like one of the things that I did when I did the actual IRB for the clinical trial that we are proposing in alpha particle RPTs for radium-223 was I took this data
And I presented that to the committee and I said, look, these methods are reproducible. That's why I think our method should work. And I think they got convinced by that too, that yes, they should work because
So
of
allow you to do the real clinical,
allows
real patient study.
yes.
I think, yeah, yesterday when you told me this, I was like, yeah, so it's like, it's not just about showing evidence and getting a beautiful publication, but it's also to have more leverage to convince clinicians to also now go to work with you and to actually do the studies.
To do the studies, right? And I really had to rush through the work that we did on CT-less last time. Same story. We did a model observer study, which was like a little bit like a virtual imaging trial because you had model observers and not actual human observers. And we showed with those that our methodology was showing promise. And so then I went to my physician collaborator and said, look, guys, this method works because I have it with mathematical observers that emulate you. And what we are seeing is that we are observing our methodology to give good performance. Now, can we get your time? And I thought that they were then convinced, but I was also more convinced and more confident as I asked them. That is why it is very important that this trial be prospective because the first person you're trying to convince is yourself, right? And if you go through the clinical trial documentation, it's a lot of work, right? I think that in some sense, when you know that the study has worked, you are saving yourself work. If this does not work, you know that it's not going to work. So you actually save yourself more time later on so that you can work on maybe a different project rather than, So being true to yourself actually is valuable, not just to the community, but to yourself because it saves your time of all the times. So then comes, any questions on this part? Yeah, this is, I think this virtual patient generation, I would call it as the most tricky part because here you have to make assumptions and you have to feel good about those assumptions. And because you will not know everything about the patient. And in the case of PPK, you'll have the same challenges. So yeah. But after that, I think is more comfortable territory, at least for me, because I have some knowledge of SPECT physics. So I can do the SPECT imaging of systems and we do Monte Carlo based approaches. To your question of exclusion. Yeah.
Why will alpha hydrogen be high energies by using the lower energy and the medium?
I will come to that question. I just want, because that's in this imaging part, I want to come to Farishta's question about exclusion criteria. Right? So when you do an actual clinical trial, you have to exclude patients. So you will see here that we did these sampling of diameters of the lesions from a Gaussian distribution. And what do we know about a Gaussian distribution? Sometimes the values can be less than 0 even, but sometimes they can be low. And so we set an exclusion criteria that, if the lesion is below a certain diameter, we are going to exclude that lesion. And so with that, we were able to, we excluded and there were some other things as well. Look at the patient and there's some computational fault as well sometimes, but mostly this was our exclusion criteria. And in the JNM paper, we describe in more detail, how we went about doing that. So we had these 12 lesions whom we just exclude, or 12 patients whom we kind of exclude.
Like the computational error was below the curve.
What I'm speaking about is basically sampling. I want to use the word artifact, but when you sample from a Gaussian distribution, you inherently get values that are low. And this is like a patient who's coming in who has such a small lesion that they would not qualify or not be eligible for this trial. So again, you define eligibility criteria, you sample a lesion size and sorry, you can't be recruited. So, but that's a virtual patient, would not mind.
How do you differentiate between failed insertion and the computation?
And so then some of these cases are right. There are computational errors. We have XCAT phantom. Sometimes the tumor is too large, and you're trying to put that in the bone, and then it just becomes does not look like a real tumor anymore after that because the tumor is large and the bone is like this small. Right? So
you do go through every individual patient.
Yeah, actually, my student.
That's interesting too, because often we generate lots of data, and in our lab, as we talk with different people in our lab, I always emphasize, as imaging scientists, we have to look at images.
Oh yeah.
Sometimes we run stuff, we never look at images. Right?
Yeah.
So that's why it's important to. But in your case, you're literally going through all the images.
Yeah. So we try to actually go through the images. And I want to say here that this lesson actually Arman had taught me once that go through the images. We were doing this no gold standard evaluation methodology, and I was very early in my postdoc at that time with Eric, and then I showed Arman kindly offered to help me with a presentation. I put together a fully 3D meeting on no gold standard evaluation, and there was one case it was not working, and Arman was like, "Why is it not working here?" It's like, I don't know exactly why it's not working. It's like, have you looked at the image? I'm like, no, not really. That thought did not come to my mind. So Arman said that, you can look at the image and that would provide you with some insight. And I have, that learning experience has been really very valuable and I keep telling that to my students now. So, that, you should look at the images. You are an imaging scientist, and sometimes the images tell you much more of the story than some data point can tell you, because our data is not just data, it is image. Right? So, okay, so now coming to your question regarding the Cheng Wang question regarding the collimators. So what we wanted to do was, we wanted to again simulate, things that can happen across different centers. Now, some centers would have LEHR, but there are some centers that may have MEGP collimators and some that may have HEGP collimators that are using for imaging. So we wanted to have different kinds of scanners and different kinds of collimator configurations to assess how reproducible our method is to a combination of these scanner collimator configurations. So we had data from let me see if I can show that here. So we have data from 3 scanners, GE, Siemens, and Philips, so 3 different vendors, with each of them having all 3 kinds of collimators: the high energy general purpose, medium energy, and low energy. Now you're correct that, with radium, oftentimes you are wanting to do imaging with the low energy, but we wanted to assess how reproducible our method is if there is a certain center that actually uses MEGP and does not use LEHR collimator. Right.
That's good. Hold on, because it's if you're using the real same collimator, that it's a high temperature, will be your fast, completely will be gradual.
Exactly, and that is the goal. Even in that case, is our method reproducible or not? Right? If I have a different collimator that, that may not be ideal for that setting, but someone uses that collimator, how reproducible is our method going to be in that setting? So, yeah.
And how about processing? For example, maybe these different vendors use different methods for scanning their
Yeah. So in our case, this is the acquisition part. There's again the processing part after that, which we compare different processing methodologies. So we have our own method, which is LCQ SPECT, but then we also compared with OSCM and OSCM with GTM based correction. I'll get to that actually in a minute. Yeah. And I should say here that I don't have his photo, but Zekun Lee actually was the one who really sort of led this trial, did a lot of the legwork, looking at the images and seeing that everything was good. So he's now at United.
Oh,
yeah, he's on the task force. He's on the task force as well.
oh yeah, he moved to you. Okay, yeah, he
Yeah.
graduated. That's right. That's right.
So, so this is your question. So now we have the projection data, and what do? We want to assess reproducibility. That is the primary objective, but there are secondary objectives too. Secondary objective: how accurate is our method? How well does it compare with conventional methods like OSCM based methodologies, right? And these OSCM based methodologies will have their own scatter correction and their own attenuation correction. Our methodology, as I mentioned, is getting a system matrix using CEMENT, so it's Monte Carlo based approach to derive the system matrix, and then an iterative technique to estimate the activity uptakes in the VOIs. In contrast, these OSCM based methods, they are we again try to apply attenuation correction. From what I recall, we also did scatter correction with, I think, triple energy window compensation techniques, but I'll have to look back at the paper to see if that was indeed the case or not. So, and you will note here that we looked at dose estimation, but we wanted to look at estimated dose and not just estimated activity. But because it was alpha, we could use a single time point dosimetry model, which could take the activity and give us the dose values. So estimated uptake of lesions, I can see here, and these are the OSCM reconstructed images with the different scanners. And you can see the variability that's there in the different scanners with the OSCM reconstructed images. And you can see the variability obtained using our LCQ SPECT as well as the OSCM for one given patient, this lesion. So you're already seeing that our methodology seems to be more reproducible compared to the OSCM based methodology. But this is just one patient, and never make inferences from one patient. You have to look at your population, right? Because you never know. It's just a sampling error, really. So
Like that, why are you showing us just a single patient?
Just to give you a representative case, yeah, not to convince anyone.
Okay. Okay.
Just to give you a visual feel in some
Yeah.
sense. You can see that there's variability here, right?
Gosh.
This is what I want you to do to convince you and my clinical trial folks that we should do this, which is looking at intra-class correlation coefficient of estimates of activity uptake obtained using our LCQ spec technique, and you can see it's only on LCQ spec. What we did was that we got the estimates and we got the confidence limits on those estimates, which both the 95% upper and lower limit. And what we observed was that our methodology was yielding an intra-class correlation coefficient, which basically measures, if you think about it, right, just it's sort of measuring how correlated it is across different scanners. Values greater than 0.75 demonstrate good reproducibility in previous literature, and we found that our methodology was giving a ICC of 0.805 with a lower limit of 0.79, showing that our methodology was reproducible across these 9 different scanner collimator configurations. Now, at this point, I'm showing you the primary objective findings. If you have any questions on the trial, I welcome that.
Ask, but I don't know. We have 30 minutes.
Oh,
So I
okay.
want to make sure you
Sure.
also get to the other studies.
Okay. Well, then we use the coefficient of variation to compare the reproducibility. Now here we're comparing reproducibility of LCQ spec and OACM based methods using the coefficient of variation. Again, we find that our methodology across a range of lesion sizes and across different organs gives a higher reproducibility compared to the OACM based approach. Here, I'm not showing you results with the retest studies as much. These are there in this publication. I'm also not showing you results where we looked at the statistical distribution of the activity values. Again, they are there in this publication. So the conclusion that we got from the trial was that our primary endpoint was satisfied. So our low count quantity spec method yielded good inter-scanner collimator configuration reproducibility for dose estimation in radium 223 based alpha particle radiopharmaceutical therapies, highlighting the potential of this for dose quantification in alpha RPTs and motivating further multi-center clinical evaluations. So this is one of our trials. This in silico imaging trial space has multiple applications in nuclear medicine. And I'll try to show you some of the other ISIT questions that we've tried to answer through this ISIT metaverse. So I actually showed one of these yesterday, so I'm not, where we asked the question that, how does task-based assessment compare to evaluation of AI methods using RMSE and SSIM? So not going to that, I want to talk about some of our more recent work. We did objective evaluation of an AI-based method for transmission-less attenuation correction for dopamine transporter SPECT. We wanted to assess how accurate our method was, but the primary objective was assess, and you can see it here, sorry, it's written in a little bit small font, so I'll try to read it out. Assess, so we had developed this AI-based methodology for doing CT-less attenuation compensation. This is for patients who are receiving dopamine transporter SPECT. And we want to see how well does this compare to a CT-based attenuation correction, right? Because the goal is that, if it correlates very well, then that provides us confidence that if we don't have a CT, then with this methodology, we would be able to get similar performance as we would have gotten if we had a CT scan. The task is looking at activity estimates, how accurate they are, how correct they are. So the primary objective was assess whether the correlation between activity uptakes obtained between our approach, which we call as DAT CT-less, and CT-based correction, CTAC, as quantified again using the intra-class correlation, was higher than the corresponding correlation between the uniform attenuation correction, which is what you use when you don't have a CT, and CTAC. So
I think you might be able to zoom in if you do this.
Yeah, no, you're right.
Sorry about that.
No, it's fine. Actually,
Details of
yeah. So you can see how we are defining our primary objective. Again, defining the primary objective is very important. You want to define this in a very quantitative way, right? So that you are able to define your measure of success very clearly. So how do we define our success? Our success is, let's say there is a scanner that does not have a CT scan. You can do two things. You can do CT-less, our approach, or you can just do uniform attenuation correction. So the primary objective is framed as, if you use our approach, are you going to get better correlation to CT-based attenuation correction compared to if you use the uniform attenuation correction approach? So now you have a very quantifiable measure of success, and you are able to do a power analysis based upon this measure of success. I want to also say that we had, also highlight here rather that we have the secondary objective, which excited me a whole lot. Now, in patients, some of you, I'm sure, are familiar with Parkinson's disease and the use of dopamine transporter imaging for this purpose. So what happens is that what you're trying to do is use these activity uptakes obtained from the caudate, the putamen, and the Glos apparatus to be able to say, does the patient have normal striatal binding ratio versus do they have abnormal striatal binding ratio, right? And that is the metric for making the clinical decision that the physician wants to make. So it's not really whether your accuracy is 90%. It's really whether you are able to make that clinical decision more accurately or not if the patient has normal versus reduced striatal binding ratio, because that can change patient management. So, right, if you're using something as a biomarker, you have this clinical decision that you're making using something. Now, we have CT-based radiation compensation, which is currently the imaging method that's used to compute this striatal binding ratio, right? And with that, you are able to ask the question, are patients being well separated or not? So we asked that if I don't use CT-based, but use our approach, which is CT-less, right? Then how well can I distinguish these two patients? So asking the actual clinical question, not just the quantitative question, but the clinical question of whether you're able to separate patients based upon whether they have normal versus reduced striatal binding ratio. So this was our secondary objective, and I'm really delighted to report that with our methodology, we were able to get similar values of the AUC, which is the way that you would quantify, performance in terms of classifying, as we would have obtained with a CT-based information correction approach. Now you will note that this was a secondary objective, so our study was not powered on this objective. Our power was based on the primary objective. So when we chose the number of patients we did, which was
For power analysis, do you guys work with a statistician?
Yes, yeah, a biostatistician who we reach out to, and we have meetings with them, and we tell them that this is the clinical trial that we are trying to design.
Is it collaborator or like university service?
Actually, we have a collaborator. It's university service too, but this collaborator have had a good relationship with them. So.
Do you need them, or is kind of like formality?
So initially I felt like I could do it. But then once you talk with them, right, you realize that there is so much rigor that they bring to your study, because they think about things that, could go beyond what you have. It's very interesting, right? We are in the imaging world, right? So someone who is not in imaging, they don't think about noise and system resolution, those things as much. They're like, oh, that's an image. Yeah, that replicates what we have. But when they ask us that question, we are like, oh, no, you need to account for noise, because if you don't account for it, you'll get misleading findings. The same way they are sort of so deep into that biostatistics literature that they are able to provide. At least that's been my experience.
We have a medical oncologist collaborator who keeps pushing us to work with a statistician, and because we do AI work with them, and we my usual response to them has been, listen, when we do AI, like the people here have learned, how to do statistical analysis, kind of understand this.
Yeah.
But she keeps pushing back. And I think you're making a point that maybe there are some nuanced considerations that we may not be aware of, especially for like designing clinical trials and things like that.
Yeah, initially I would feel like, we could do it, but then as I worked with them, I realized that there are several subtleties that they are familiar with, which sometimes may not be familiar to us. Like, the kind of, again, like looking at ICC, right? How do you know, based on a finite sample size, what is the ICC that you want to aim for, and how do you know how to set your confidence intervals, and based on that, set up your sample size. So
you sit down, you organize a session for an hour, you guys go at it, you present your.
So yeah, the way we do this is actually presenting our study design, talking about our primary objective, speaking with them about what you want to do, and then after that they suggest some ideas, and then it's an iterative process afterwards where they get back to us and say, oh, this is what we have. Now it requires us to become familiar with their methods as well. It cannot, our observation has been that it cannot be a black box, because we know our trial the best, right? And sometimes they may misinterpret the trial objective, and then they may give us something that may not exactly be right for us. So it requires going a little bit outside what we know at times, which is a good learning experience as well. So yeah, but yeah.
One question I have, I guess the number of patients from this analysis is like the lower limit, that at least you need to make fine confidence with that you
If-
can demonstrate.
If an effect is present, then you can see the effect.
Yeah,
Yeah.
I guess the question is, what stops you from simulating more, like having a longer component?
Yeah, and again, because of the idea we want to emulate an actual clinical trial, so we just say, okay, this is what we can. Some cases, and there are other cases where you are also limited. Like for example, this virtual imaging trial was really based on clinical data, where we were having the clinical data obtained from patients who had MR, CT, and PET. And so what we were able to do with that was define these boundaries of choroid, putamen, and Gros paratus, and then assign activity uptakes. Because at the end of the day, our goal is to be able to see how accurately we can quantify. But we kind of took real data, right? And that real data where a patient has gotten these 3 scans or 2 scans is not as many. So in this case, you will notice that you had 197 participants who underwent tri-modality PET MR CT. That patient number can be somewhat on the lower end of
So you
that.
don't do like Gaussian sampling, right? You actually assign real patients
In this case.
in this one.
But in the ISIT QA, we did not do that. ISIT
For
QA
that
was
one, you could easily do.
what Pedro was mentioning. Yeah. So here, on the other hand, we have this design constraint, and so then we talk to the statistician and we say, what is the kind of power that we can get with this sample size that we have? And then, so therefore, it's sometimes an iterative process, and you have to just try to, and sometimes that guides what is your primary objective and what is your secondary objective, right? Because we may not have enough power to be able to separate patient populations, but we may have enough power to be able to say whether we're measuring things accurately enough or not.
In our lab, once we did an exercise where we had everybody sort of write down what kind of clinical trial they would like to do based on their work, like AI. But that was for clinical, but we could certainly do a similar thing here.
Similar thing, yeah. And you will note here that that we compared our methodologies with other methodologies as well. Now, as part of this, what happens is that you do the study, and there's another experience that I share, that you do the study and then you go to the reviewers, and the reviewers say, oh, I would like to see this study as well, right? For example, one of the reviewers wanted us to compare our methodology of DAT-CTLS with other deep learning based approaches. We had compared with one, but they wanted to see a comparison with something else too. That's fine, but that then becomes a secondary objective. It cannot be your primary objective. You have to stay true to your trial. And you have to tell the reviewer that, look, I can't make that my primary object, as much as I would, I want to appreciate your remark, and maybe that method is yours, but, but I cannot make that. And this paper got published, so I think that they were okay with that. So you can, but with secondary objectives, you can really have a large number of them, right? You can assess test, retest, you can assess generalizability, you can evaluate your methodology using fidelity-based metrics, you can do comparison with other methods and all that stuff. So there is some flexibility in terms of things that can be added, but you have to stay true to your primary objective. Now, if sometimes you may be familiar with this, that in clinical trials, you also have something called tertiary objectives, right? So trial gets done, you look at the data and you want to use that data for something else, and that becomes a tertiary objective. So we have this data, we are very open to collaboration. If you want to do a tertiary objective with this particular study, then very happy for, and this is going to not require a material transfer agreement, hopefully, because this is in silico data. This one may, by the way, but other studies like our ISIT QA, I think should be okay. Now, we had developed this methodology CTLS. Again, I apologize that I had to rush through that methodology but it was good because I had all these discussions with you, and I really enjoyed that. But we developed this methodology for attenuation correction in myocardial perfusion SPECT, and the methodology was again showing strong promise across 2 scanners. We assessed generalizability across 2 scanners with real clinical data. We got good performance. But that led to the question, how generalizable is our method if it is actually across multiple scanners from different vendors? Again, the same question that, how generalizable is our method across a GE versus a Philips versus a Siemens. Now we have only GE scanners on our site. We wanted to see whether this would work with a Siemens or a Philips. How to do that? Well, in silico imaging trial. You don't have to take the patient and again move them between one center versus another where they may be different scanners. So we were able to use an in silico imaging trial to also assess the generalizability of our deep learning based attenuation compensation method for myocardial perfusion SPECT. This work led by my PhD student, now actually He's a postdoc at Mayo Clinic, was the recipient for the Best Student Paper Award at the SPIE Medical Imaging Conference. And we trialed it as, is it generalizable? Right? Then here is another trial that we did, which I think is quite relevant. So this was in the context of developing strategies to personalize image acquisition protocols. So James, the work you mentioned yesterday about being able to optimize those strategies, right? So we were thinking about the question that can we optimize acquisition protocols based on the patient's body habitus to find out if there are certain projection views where you want to spend more time versus other views where you want to spend less time, with maintaining the same time budget depending on the habitus, the size of the patient. Because if you have a female patient, for example, with large breasts, you have more attenuation artifacts. Patients who are obese, you have more attenuation artifacts. So it may be possible that for those patients, you may actually have different protocols. Now, the tricky part here, as we were thinking about, we did some initial validation studies with, again, simulations. Our methodology showed a lot of promise. So we said, let's do an in silico imaging trial. Now, this was tricky because this was a personalized trial, right? So it's not a trial where you are having the same protocol for all patients. Here, you can have a different protocol for each patient, right? So you want to personalize, and this is, I say this because, in the context of radiopharmaceutical theranostics, right? If we are thinking about personalized dosimetry, then we are kind of thinking about how do we optimize the patient treatment regimen based upon things that we learn about the PVK models, or from their initial PET scans, right? And then for different patients, things will be different. Now, how do you validate that? Because you can't have two populations now, right? Like, oh, there's a patient population who is just like your usual patient population getting drug A versus patient population getting drug B. That's not how it is, because now you are having like a range, a spectrum, if I may call it, of different patients who are receiving different kind of treatment regimens. Same thing here as well.
Yeah. I mean, we should. The VTT opt trial, where we're optimizing how much you're injecting into the patients based on the digital tool that we're building, coupling the PBPK with the PD and seeing if the patients are, I guess the primary objective would be to see if patients respond better to therapy compared to a one size fits all.
One size fits all.
Primary, I don't know what the secondaries are. We'll figure that out. You're inspiring us. So write it down and do it.
So
that was a pretty convincing study.
One size fits all is right here, uniform. So this was our uniform protocol. There's one size fits all.
Yeah.
So what we did was that we took a patient population, we calculated our protocol, our optimized protocol. We imaged the patients virtually using the conventional protocol as well as our protocol. And then our protocol is personalized. The uniform is one size fits all. Then we reconstruct the images and we study the power of the study to the primary objective of comparing the pre-spec versus the one size fits all protocol for defect detection tasks through ROC analysis. And from our results, we observed that there was meaningful improvement in performance. So this work was at the SNMMI annual meeting that we had an abstract on this. I'm happy to share the link to that, which describes
personalization of acquisition or reconstruction?
Acquisition.
So the duration of the scan or like what?
Duration of the scan is same. What you're trying to do is optimize the amount of time per each view. So let me see.
That changes the duration.
Yeah, different projections have different timing, but at the end of the day, the timing of the.
The total time is fixed.
Total time is fixed. So if I can show that here.
Oh, interesting. No, I wasn't aware of this one. Okay. Anyway, I don't want to distract you.
I can go through quickly. So what you do is, I can go through this. So you maximize this thing called total detectability, which if I can, this is the one, right? So you can see here that your conventional protocol would look like this.
Yeah.
You're spending the same amount of time. Using our protocol, you would find that there are some views that provide more information compared to other views. And the way we optimize is using information theory-based concepts. We try to maximize detectability, and we try to find out what is the amount of time spent in each view that leads to maximum detectability for the patient. And you can do this because you can take the CT scan of the patient, so you know what the body habitus is, and so therefore you can find out what is the amount of time spent which will lead to increased detectability.
How many angles?
So this was conventional myocardial perfusion spec. So
That's a high dimensional optimization plan.
high dimensional, as in you have to optimize with, I can show you the problem. Yeah, there it is. So you have this vector T, which you're trying to optimize such that the sum of all T is equal to a fixed time budget that you have, right? And so how many ever views you have, that decides here. So it's high dimensional in T, but T is a scalar. And all the T's are just, just
Any star.
depending on the number of views you choose here.
Interesting.
Yeah. So, and so, actually,
According to that,
Yeah.
are you giving weight to the status from the panel to include standards?
so in this case, we actually had scatter data. We did not do scatter correction. We did attenuation correction from what I recall. It was cardiac SPECT. So we're trying to emulate in the clinic that we were having, we were seeing was mostly attenuation correction that was being performed. But scatter correction is certainly an important thing which needs to be accounted for. But can you tell me the motivation behind your question?
Actually, we said different times as as age projection.
Yeah.
So actually, when you are gathering more data and more signal from this source, actually, because of this scanner distribution around the patients, at each angle we have different problems with scanner.
Yes.
So this actually might be somehow scattering at this angle might be more emphasized in still interesting.
That is true. That's why we did model scatter in our simulation studies to ensure that, again, that we are able to account for that. Yes, that's a very good point, actually. And that's what sort of formed the basis of, not formed the basis, we certainly want to model scatter, but we observed that in spite of that, our methodology was yielding performance. One way I can think about this is, if you don't mind, I can talk about this later on.
Sure.
Yeah. But one way I'd like to think about this is that, if you are sort of in those views, which are sort of closer to the heart, right? Then remember that scatter is mostly Compton, right? So the probability of the scatter is proportional to the activity and sort of proportional to the distance as well. So we did some analysis at some point of time trying to look at how much scatter happens. And of course, as you go farther away from the activity values, the amount of scatter also is impacted. So I have to think more about your question and try to understand why that could be, how that could be an important concern. I'm sure it is an important concern, but I'm not able to give you an exact answer right now because I'm not able to fully understand the concern. Yeah. But again, we did the scatter modeling here in these experiments. Yeah, sure.
So the primary objective here is comparing the detection task or analysis that you're optimizing detectability just for the first line protocol.
Mm-hmm.
Maybe I missed how you did that, but it's same model observer.
That's excellent question. So can I go into a little bit detail?
You got 10 minutes. You decide what you want to do.
Yeah. So yes, so was there's an area I find exciting, so I will take some time of my own to do that. So the way we do it is, and hopefully I've been able to give you some arguments on why we need to do it. So, why do we need to go from one size fits all to more personalized imaging? So there is this thing called an ideal observer. And ideal observer is an observer which tells you what is the amount of information content in the data, which observer that is the best possible observer that there can be. It's a mathematical observer that is the best possible observer that can be, that will be able to perform the detection task. So you are optimizing for that observer. And the reason you choose that observer is because you want to maximize the information content in the projection data, right? The underlying hypothesis is that by being able to maximize the performance, maximize the information content in the projection data, you would therefore also maximize the information content in the reconstructed image. Reconstructed images are read not by ideal observers, they're read by human observers. Human observers are suboptimal. They are not as good as ideal observers. And as a result of that, therefore, it is important to be able to quantitatively assess whether this maximization of information in the projection data translates to meaningful improvement in performance on the reconstructed images. with
For studying the reconstructed images, did you use actually human observers? Did you use like a
anthropomorphic model observers.
models that
Yeah.
are not ideal?
They're not. But we also did human observer studies, not statistically powered. These were just, showing them samples. Yeah. Showing them some views and asking them the question, is it or is it not? And this is work in progress at this stage. So there are some initial findings which are very exciting to us, and we hope that we can get support for this, and then that can help us expand further on this work. But this is some very exciting stuff that shows that yes, if you maximize the information content, then you can actually achieve better performance in the.
Isn't it that the anthropomorphic model observer is only, you're able to use it image based, you can.
Yeah, exactly. So you're maximizing the information content, the projection data, with the hope that, or with the assumption, and I think it's a reasonable assumption. If I start off with less information, I'm not going to synthesize new information, right? So let's start with the data that has the most information, and then do the reconstruction. And the idea is that the reconstructed image will therefore have more information.
Is there a way to get that model observer in projection space?
It is tricky, because an anthropomorphic observer kind of works on reconstructed images. Human observers don't look at projections, right? They look at reconstruction. So if you want to emulate a human observer, you have to do this anthropomorphic model observer. As much as I would actually like that, but if you have to do clinically relevant study, you have to work with the human observer. That's good. Yeah. Okay, so yeah, so that's perfect. So we did another work that we are very passionate about. This idea of no gold standard evaluation. I talk and we talk about task-based evaluation, but we realize that one important thing for task-based evaluation to happen is availability of methodologies that can help us do evaluation with clinical data. With clinical data, we don't have ground truth. So how do you do evaluation on clinical tasks when you don't have ground truth? So let's say the task is quantifying, tumor volume. I don't know what the true tumor volume is. Right? And I can have a physician draw a tumor volume for me, but the physician defined tumor volume may have its own source of errors. So then how do we do evaluations when we don't have ground truth? So, Farishte comes up with her segmentation method, and then there are two other competing segmentation methods, and she wants to show my method is the best. She uses clinical data for that purpose. How does one show as an investigator that my methodology with clinical data works well? So for this purpose, we have developed these statistical no gold standard evaluation methodologies that are based on the assumptions that there is an underlying stochastic relationship between the true and the measured value. And so if I could estimate the parameters that describe this relationship, I would be able to say which method has more bias and which method has more noise. Now, if I knew the truth, I could do it right away. I don't. But that methodology, what it does is that it assumes the truth is sampled from some distribution, and based on that, it is able to identify which method is actually having higher amount of noise. And so therefore, it is able to provide an indication of which method is better. Not going through into that in detail, I'm happy to talk about it. But then we developed the method and we worked on it. And again, we did several studies and it showed promise. Well, how do we know if it's going to actually work with clinical data, right? Because again, you don't have the ground truth. So how do you validate this validation method, right? And so therefore, we ask the question, is it right? Is our method actually able to correctly rank the different methods? So virtual imaging trials can be used for that purpose as well. So we developed this method, which we call as NGAC CORE, no gold standard evaluation, CORE referring to correlated noise between different methods. The primary objective was evaluating the accuracy of NGAC CORE in identifying the most precise method. This was one trial where I think talking with the biostatistician was very useful, because we actually did a lot of numerical studies in order to identify what is the size of the patient population that we need in order to assess whether our methodology gives accurate rankings, because rankings are a nonlinear function of the activity value. So it was a lot of simulations that we did to do the power analysis to identify which method.
What's the task? You're evaluating what exactly?
We are looking at ranking performances. So we have
Ranking of what?
three methods. So this is in the context of, again, alpha particle RPTs. And so we have these patients who are scanned using using a single scanner. And then we are reconstructing them using three different reconstruction approaches, one of which is an OSCM approach with attenuation correction. The second one is an OSCM approach with correction for attenuation, detector, and scatter response. Sorry, scatter and detector response. And then you have this methodology, LC-Q-SPEC, which is
It's a detection thing, or well, you're comparing their performance in
On the task,
Estimation?
yes, estimation, yes.
Estimation of like
Activity,
lesion or
yeah.
activity uptake. in some
Activity
organ?
uptake in organs. So this was organ dosimetry. So we are looking at
Organ dosimetry.
organ dosimetry. And we are asking the question that can we rank these methods accurately? And we know the true ranking because it's all in silico. We know what the true ranking is. And so the primary objective is, can we rank the methods correctly using our methodology? So to ask that question, we need to have a sample size, but ranking is a nonlinear function of the data itself, because ranking is like, you get a number, but then that maps nonlinearly to rank number 1, right? Someone gets a 90, another one gets a 10, that's rank 1, rank 2. Right? And someone gets a 90 and another one gets a 50, and that is also rank 1 and rank 2. So we had to do a lot of power analysis to be able to identify what was the right number of patients. And this is where one of the
It's very complicated.
it was very tricky.
The statistician even had to learn stuff probably to do this, right? Or was this an obvious problem for them?
Yeah, I don't know. I know it was not obvious for them. So I think that they kind of like this research, so they really helped us out with this. We did, and we did a lot of simulation studies on our end, so as to be able to see whether our method was where we were able to collaborate with them. This work was led by Yan Liu, PhD student in my lab, who is getting close to graduation now. And at the recent SPIE meeting, she had 2 talks on no gold standard evaluation. One was comparing no gold standard evaluation with reference standard. Another one was developing a new no gold standard evaluation technique that uses data from some small number of cases where ground truth can be available. And for validating both of them, she did actually in silico imaging trials again.
So for AI, it would be like the equivalent for us would be, imagine we have 3 different AI solutions for
Yes.
predictive modeling.
Yeah.
Or whatever, but we don't actually know the outcome for the patients.
Yeah.
Can we develop a technique and validate that technique that allows us to know which of the AI techniques is better, even if we don't have the reference truth?
Yeah.
For segmentation, that certainly makes sense, because for segmentation, even clinicians sometimes don't know what is the exact.
Yeah. So one thing which I want to remark here is, add as a qualifier here, is this methodology specifically for quantification, right? So it's about being able to validate methods that provide some underlying quantitative parameter of interest. There's some work also done in the area of using NGSE for classification-based approaches.
But so we could, let's say we have a model for TMTB.
Yeah, exactly, right? And you want to say that,
We throw it at the 1,000 patients.
Yeah. Like you want to say
without reference truth delineation.
Right.
We might be able to know which one's better.
Yes, and actually what we found, and this would be of strong interest, I think, to Farishte and to several of you as well, was that sometimes you do physician delineations and you think that's your ground truth, right? But you know that is inaccurate, right? What we found was that after a certain level of error, actually physician delineation defined evaluation performance is lower compared to no gold standard evaluation performance. That is what the finding was presented by Yan at SPIE. He actually also,
as people move towards like unsupervised AI and all those things where you have less and less reference truth delineations and reference truth anything.
Yeah.
I still have a philosophical question here. I don't want to waste your time, but I think, for example, the clinical trial, we have this exclusion criteria, and you list some of the region and some of the patients because of the exclusion criteria. But what limits your university to reverse the step 2, for example, and come back to step 1 and add those cases that you rejected already? The same number of cases.
Nothing, actually, you can do it. It's just that you don't want to, because you want to do it purely prospective, right? You don't like I mentioned at the beginning, right? You want to get confidence from this data, right? And that is why prospective study is such a gold standard in clinical trial space, right? Because we know that you cannot go back and recruit patients because something didn't work out. So this is like you can make it really prospective, and once it works out, you will be sky high. It's like, yes, this thing really works. So with that, let me present to you the ISIT metaverse. Where I'm presenting these 5 trials. We have also done other in silico imaging trials. We find them to be a very valuable tool. We have done stuff for validation of our quantitative spectrum methods for thorium and radium quantification. We have also developed in silico imaging trials to assess whether task-based evaluation is even needed for AI-based methods. We find this to be a very useful, reliable, safe, cheap, and ethically not questionable, logistically very appealing tool, and very happy if you have any questions about these tools. And with that, I'd like to conclude that virtual imaging trials can provide a safe, inexpensive, fast as in fast, relatively fast. It's well, it does take time to do all the simulations. So it's faster, probably is the correct word, compared to doing an actual clinical trial, and reliable mechanism to perform rigorous early stage evaluation of imaging methods for nuclear medicine and theranostic applications. And with that, I have to really acknowledge this is truly, like a clinical trial. This is a collaborative effort.